Konnichiwa. Hi. Welcome to the last session of Tokyo ALife 2020. And the final session is Symbiotic View of Life. Okay. So first of all, let me explain why I choose this theme. One of the reasons I have chosen this theme is because of this coronavirus. This conference was scheduled on 27th of March, but I couldn't make it because of this coronavirus, right? So we postponed to 18th of September. But again, we can't do this because we can't invite people from United States or Europe, right? However, this VR environment is I think it's very interesting and challenging. For the last session, it was about VR. VR and AR can be the next tool, and then also a platform for studying artificial life. So I think we are taking this opportunity to invent something, right? To learn how to deliver our ideas and our messages to the people through this VR and online conferences. Okay. Then let me introduce why I asked for these 5 people to join this session. So Dr. Tom Froese, he's from OIST in Okinawa Island, and he has been interested in life and mind continuity. I will explain that later. And next, Dr. Hiro, he's from Hokkaido University. And I have been working with him for many, many years, like more than 15 years now. And then he's the guy that he invented the concept of homeostatic adaptation. Next one is Dr. Olaf Witkowski. And then he's originally from Brazil. And then he was studying computer science. And then he came to my lab. And then we did some swarm behavior, swarm dynamics. And each agent has a neural network inside, which is, I think, it's one of the few research who invented this new model that it's not an empty particle, but particle with inside internal state, and then it's swarming around with each other. Also, Professor Ichihashi Norikazu, that he's also from University of Tokyo. He's my colleague, but he joined us from the last year, and he has been working with Tetsuya Yomo in Osaka University, and they found a very, very interesting experiment. And also, he made a big progress this April, and then I was really wanted to invite him to explain what his discovery is. So that's why that, and then I don't think he's himself called artificial life researcher, but that's exactly what artificial life was aiming at the early stage. And the next speaker is Dr. Takeuchi Hirotaka, and he's from Polar Chemical Research Institute. And he also doing a very interesting work. He's not only studying biochemical experiment, but he tried to make a relationship between biochemical microbes and our beauty, right? Cosmetics, which I think is another future application of artificial life. So let me introduce you some of the works which is more practically connected to the session. The left top corner is one of the first work I did with Tom, which is about oil droplet in computer simulation. Not only 2 chemicals. 2 chemicals is a minimal number of chemical components which generates a bunch of interesting patterns. But once you add a 3rd chemical component, you can have an oil droplet like moving glider, like this one, as you see here. And the right bottom corner is something is moving around. That's the oil droplet that I found with Martin Hanczyc. He's from Italy. And then this is the oil droplet that I found in 2007 or 6 under the microscope, that usually the chemical reaction is going into the large space. But this one is localized in 100 micrometers, and then it starts to move around spontaneously, which I found was very interesting and then very exciting. So that's what I became very interested in the real life. It's not life, by the way. So this is a pseudo life made by the chemical reactions. And then top corner is the collaboration with Olaf, that he's evolved. It's not empty, as I said, agent with an artificial neural network inside, and then each other is interacting with each other, and then gradually, I mean, after a few thousand steps, then it becomes swarming each other. So we are interested in what kind of neural network inside the agent that enables the agent to cooperate each other or swarming each other. That's one of our collaborative work, which was published in 2018. And then the middle bottom picture is a little bit abstract phase diagram, but this one is Hiro Iizuka's concept of homeodynamic plasticity. So once your agent get injured, but he tried to repair by himself and then moving around again and again. So the homeostatic or self-repairing system is one of the inevitable and then one of the most important property of the living systems. I think he's going to explain that in detail. Or it doesn't have to be in detail, but that's what I want to hear from him. Then middle upper figure, which is a bit complicated, but this one is, I think, one of the first example that evolved RNA sequence is like a species, and then it is dividing into one species and another species. So Ichihashi sensei is projecting onto the 2D space to see how this speciation has been happened in even RNA sequences. So this is not, again, this is not a living systems, but this is a pseudo living systems which has the RNA sequences like coronaviruses, right? And then last, the left bottom corner is Takeuchi-san's experiment, which is even aged cells. If you reactivated one of those RNA sequences, which is link 00942, then this cell get younger again, which is one of our dream, right? How you get young again, right? And then he is trying to find out a hint for doing this. So these are the people. These are the works that those people that I invited today has been done for last decade, I think. Okay, welcome to the session. And then one of my question to you guys is, what is your open problem in your research team? Okay, so can we start from Olaf?
Sure. Thanks for having me. I guess the important open question would be the question of openness or open-endedness. What we call open-endedness is this ability for evolution or systems to invent new things. So inventing forever or learning forever. So this is a question that my lab and my collaborators are asking nowadays, and I find it maybe the most fascinating question in artificial life. So if we can build a system that can keep learning forever, what does it require?
Okay. Hiro, can you... Hiro?
Oh, yes.
Are you trying to escape?
No. Well, but I just, could you, can I do it later? I just want to explain the evolution first.
Okay, Tom, can you give us
sure.
Yeah.
I'd like to build on Olaf's open problem of openness and specify it a little bit. And it has to do with the work that you introduced about the chemical reaction network gaining new properties through the parasite interaction that suddenly starts to move around through the space, whereas before it remained static. Mm-hmm. That work that we did together with Nathaniel Werger raised the question of what's the role of movement and behavior in evolution. So usually people think about evolution mainly in genetic terms. So it's, RNA sequences and DNA adapting to changing circumstances. But what about behavior and movement? Maybe part of the openness of open-ended evolution can derive from the openness of behavior and the surprises that interactions can bring about. That's one of the problems I'd like to think about.
Mm-hmm. Thank you so much. It's very interesting. I'm interested in this one too. Takeuchi Sensei, can you?
Yes. My ultimate goal is to want to know how we get agelessness. But my question is why we have aging system in our body. I hope we find a way in the point of view evolution, and I want to know the relationship between aging and evolution.
That's interesting. Yeah, thank you so much. Ichai Sensei, you want?
Okay.
Hi.
My question is also same as Olaf's question. It's open-endedness of evolution. We are constructing our RNA replication system. It's a non-living molecules, but it has the ability to replicate and also acquire mutation by replication error. So if we repeat the replication of the RNA, it evolves spontaneously. Mm-hmm. But we found that the evolution of the RNA system is ended soon. It's not continuous, but the evolution of life is different. It continues for very long time. Even now, new species appears. So I'd like to make a system to evolve continuously. That is my question.
Okay, thank you so much. So, Hiro, are you ready?
Yes, thank you. So thank you so much for the time. And then my question is also related to the open-endedness. I've been working on the artificial life simulation for a long time. And then I try to create the evolutionary system in a computer. But always I can create the one-step emergence behaviors. But we cannot create the multi-layered hierarchical evolution. We just one step bootstrap is okay, but we cannot create first and second, third, fourth, and that is my open question. And if we can create that kind of simulation, then we could understand evolution more, real world evolution more concretely, and it will help, I think.
Okay, very good. So everybody's interested in open-ended evolution. That's the main theme of artificial life study, and I'm also very interested in this one. But in order to understand open-ended evolution, we have to understand what is a replicator, what is the evolutionary system is. I think it's ordinary people never heard of self-replicators or evolutionary systems. It's only known, but Darwinian evolution, mutation plus selection goes to something, and I think very few people heard about open-ended evolution is. So I asked Olaf to introduce what is replicators, what is becoming complex, and what is artificial life study is discussing on this issue.
This is just an explanation of what is replication and how you get to evolution. And it's not so hard. So even if you never heard about this, replicator is a complicated word to say something that copies itself. So something like, for example, a virus, or maybe some RNA, something very simple, even some code. can replicate itself. And that's different from the concept that we want to talk about, this complex evolution, right? This symbiosis and so on, that creates complexity, so much interestingness in the world. So if you start from the replicator, it does nothing else than making a perfect copy of itself. This is a video from Bert Chan's linear model. And it's just replicating. So creating a copy, and that copy will create another copy, and so on. And if the copy is perfect, it's so boring. It's great, but it's so boring. But if you have some variation, if it makes actually a single mistake in the replication, then the copy is not perfect. And if the copy is not perfect, you end up with a lot of different diversity variation. So that becomes interesting because if you select the best variations or the ones that survive in an environment, that way you get evolution. And there are many simulations in the field of artificial life that try to tackle just this. And you have a few videos here below. So if you can see them from the left, you have very simple models of copy, just replicators. In chemistry, molecular models, also computer 3D models of simple creatures. And then if you have mistakes that are being introduced, then you have evolution. And even further, if you move further to the right side, you can see that there are models that we study in which you have interactions between different species, different individuals will, during lifetime, interact with each other and create cooperation or competition first, but also symbiosis and complicated interactions between those creatures. And I think that's what we want to talk about today, because those interactions have the power to create more open-endedness, interestingness, interesting mechanisms in the world.
That's great. So you came up with this word, symbiosis, yes. And then I think we have to put stress on that there is no randomness inside, right? So Bert Chan, he introduced his replicator, which is, it's not a game of life, but it's a continuous version of a game of life, if I'm allowed to say this. So this game of life is, it looks like cell automata, but it's not, because the state is continuous. But it's looking using a neural network to get the input from the neighbors and then change its state. And the interesting point is there is no randomness. It's coming from outside or inside. But the purely deterministic system becomes replicator. And then also when they are colliding each other physically, and that generates new replicators. So which I don't know whether this one naturally happens in the world, but that's what he found. And I think that's one of the, I think it's one of the artificial life's idea and concept that we don't need randomness, right? We don't need the external world, but everything is inside the system. And then the interesting evolution is coming up. However, the question is, where is the open-ended evolution is, right? So open-ended evolution that, 5 of us, everybody think the open-ended evolution is one of the open problem. And I agree with this one, because once you have a replicator, and either there is a mutation from outside or colliding each other, generate new replicators. However, this new property is somehow bounded. And we don't know why it's bounded, right? So coming back to, I forgot, maybe Ichinose Sensei can correct me, but 1970 or something that Spiegelman, Dr. Spiegelman in Germany, that he, for the first time, did using the real RNA of phages, I think using RNA sequences to replicate itself. But it's not itself because there's a bunch of enzymes and then nutrients coming from outside. So this RNA sequences can replicate himself, okay? So the question is, whether you have some, actual Darwinian evolution is happening from these RNA sequences, and then can we see some new sort of a complex something is coming out? However, this experiment didn't show any interesting evolution at all. The RNA sequences is getting shorter and shorter, and then stop it there. Then after that, so this experiment was very famous, and people were very surprised because it's not a living system. It's just the RNA sequences, but they can replicate itself. It's like a living system, okay? But didn't show any evolutionary processes. So that was a big problem at that time. But then Yomo and then Ichihashi sensei came out at Osaka University. I think they tried to do the same experiment. However, of course, that evolution didn't happen. So as far as I remember correctly, that they tried to encapsulate. They have small, area, and then each RNA should stay in these small, areas. So they are disclosed in these areas. Then also this RNA is replicating enzyme is not coming from outside, but this RNA is also coding this replicating enzyme itself, okay? Therefore, it's a sort of a complete system. So this RNA sequences is copying himself by using its own enzyme coding by himself. And then it's increasing its number, and then there's the mutations, and so there's a parasite. It's interesting. However, it's always coming back to the very simple systems that never show any evolutionary system at all, right? So it's a huge problem. Why there's no evolution at all? RNA sequence can replicate, but becomes smaller and smaller, and there is no evolutionary processes that happen at all. But after that, his recent paper, Ichihashi Sensei's recent paper in this April, he showed that so that's what I want to know from him. He did some little trick. So the replicator is coming, increasing and then repeating himself. And because of the mutation, there's a new parasite is coming from this RNA host sequences. However, they will never die out, right? The parasite coming out from the original RNA sequence is increasing its population size. And then after 300 generations of, 400 generations, still it's going on there, right? Mm-hmm. So it looks like now the evolution is possible even in a test tube, right? So I was really surprised, reading his paper. I vaguely know that, from rumor that this experiment is going on, but I was really surprised. So if Ichihashi Sensei can explain, because this one is one of the hints whether we can have an open-ended evolution in the test tube, not in the natural world, not in a computer, unfortunately, but in a test tube, it looks like open-ended evolution is happening, right? So,
okay, thank you for a good introduction. It's yes, very precise. What we did is we successed the Spiegelman's RNA system, but the difference is a translation mechanism, which is a production of enzymes. That is a RNA replication enzyme from RNA itself. So another different point is encapsulation into the system into a very tiny droplet. It's a bacteria-sized droplet. That is a very important point. Without it, RNA cannot replicate for a long generation. It accumulates mutation and died out. So as Ikegami-san said, in our system, even we introduce the translation system and encapsulation, the RNA evolution does not produce any interesting phenomena. Its replication ability increase, but that's all. No complexity appears. But when we allow the appearance of parasitic RNA is produced, as Ikegami-san said, it's produced from the original RNA, but it's lack a gene, which is encoded in the original RNA. So it cannot replicate itself. It needs another RNA in the same compartment, same droplet. In this sense, we call it a parasite. And if we allow the existence of parasitic RNA, the original RNA evolution became continuous, never stop. Never stop, I mean, even now, its evolution is still evolving, almost constant rate. I can measure the evolution rate by the number of mutations accumulated in the RNA. So, in my feeling, I think that the existence of parasite allows, I'm not sure I can say it's an open-ended evolution because I feel that the evolution is still limiting to some extent if we compare to the natural living organisms, because during the evolution, we didn't find any development of complex network yet or new functions yet. So I'm not sure it's open-ended evolution, but I can say that its evolution became continuous according to owing to the existence of parasitic RNA. So as Olaf said, coevolution is one of the key. I'm not sure it's a parasite is required or not, but coevolution with other replicator is a key function to achieve the open-ended evolution.
That's very interesting. From my experience in simulating replicators in a computer, first of all, even if you prepare some complex replicators, because it takes more time to replicate itself, so it's getting simpler and simpler, and finally, it is dominated by the most simplest replicators. This one is true for Quawos. It is the one with the early stage artificial life simulator. Quawos is you have some red code simple program, and then whether you can replicate itself or not. But finally, it goes to very, very simple replicators. So to me, it was one of the big problem is how to avoid going into the simplest replicators, even in computer simulations or chemical experiments. Right? And then Eigen and Schuster also discussed this hypercycle is one of the trick that you can have complex replicators. However, again, this hypercycle, starting from a 5 lengths, but it becomes 4 lengths, 3 lengths, and then became one of the simplest hypercycle can exist. So how to make a complex replicator is the one that it's a paraphrasing how to have open-ended evolution, I think. So I really want to know what's the hint and why this is possible. And then you said it's maybe the coevolution between parasite and host sequences is one trick that I think it's a very slight balance between something like how maybe if the interaction is too strong, then one of the hosts dominate the whole systems. But
I see.
If the parasite is strong enough, then I think it is overcome by and then it is extinguished completely. So I think this balancing between parasite and host species is something I want to call homeostatic dynamics of the ecosystems. Right. So that's why I also introduced Hiro, because he's a specialist on homeodynamics and homeostasis of artificial systems. So
yes.
Yes.
I'm not a specialist, but can I share my slide? I just prepared the ALife simulation. I would like to explain my favorite ALife simulations, which is called ALife. But this is the original long-term self-replicating loops. And I just want to explain briefly. And Langton constructed a simulation world to investigate how the self-replicating process can be realized in the local interaction, like a chemical reaction. The space consists of discretized cells, and each cell has a discrete state. And those states change depending on the surrounding cell state. And like this video, so this one starts replicating like this. So all things happens in the local interaction, like a chemical reaction, right? This is the original self-replicating loop. And nothing interesting happens here, just replicating like a crystals. And then this is the Simon-san's work, which is called the envelope. And this envelope adds a slight modification to the original model. And then the original self-replicating loops were just replicating like a crystal, but the Simon-san introduced collisions between each other. So when they collide each other, something strange happens. And then as a result, new form of self-replicating loop is created. And it begins to reproduce itself. And of course, sometimes not, but sometimes it's possible to replicate. But if the new self-replicating loop becomes larger, the probability of collision increases. And then the probability of collapse or dying increase. But if the new form is a smaller loop, then it's less to collide with each other, and self-replicating more quickly happens. So it takes up more and more in space. So when another collision occurs, a new form might be created continuously, right? And then originally, the self-replicating loop is quite big, but in the end, the self-replicating loop becomes very smaller and smaller. And then evolution happens like this graph. So at the beginning, the self-replicating loop is very big, but then becoming smaller and smaller. And then they occupy the space. So again, in this simulation, Simon just implement the chemical reaction in a local way. But in the end, evolution can happen in the simulations. And the problem is that even in this simulation, as Takashi said, but small self-replicating appears and then finish. So never more evolution happens. So I think we need some trick to bootstrap the continuous evolution. Yeah, that's what I wanted to say, and then want to clarify the problem in the simulation or experiments. Okay, Takashi?
Yeah, sure. Thank you for clarification. Yes. So this is a concrete simulation that demonstrates, even if starting from it's gradually becoming complex, this replicator, but finally goes to the minimal replicators and never coming back to the complex and larger replicating loop. So what's the problem is, right? That's what I want to understand within 55 minutes, right?
Can I say something?
Yeah,
Takashi?
of course, please.
So when we discuss our open problems, and now we dug a little bit deeper into the question of open-ended evolution and why don't we see it happening? Well, the first thing that strikes me is that it doesn't seem to make a difference what kind of system we're looking at. It can be a continuous simulation, discrete simulation. It can be a computer simulation. Mm-hmm. It can be an actual chemical system. And yet we always run into the same problem. So it seems to be a very fundamental thing that's missing here. That's not specific to any kind of implementation that we've been discussing here. But one thing that Hiro said in the beginning that resonated with me, and that is that in all of these cases, what we see is like a one-step emergence. We never see a second-order emergence. So we have some simple local interactions, and then we get evolution happening. Or we have some simple local interactions, and something grows, or something moves. So we never build these like hierarchical levels. No. But as I said in my comment, one of the interesting things is behavior. So it's usually living systems do something other than just metabolically reacting and replicating evolutionary. And also Hiritaka-san's comment about aging, I think, is very relevant here, because also development and then to some extent undevelopment or aging at the end is left out of all of these implementations. So we seem to be combining the very fast time scale of local, almost chemical style interaction with long-term evolutionary time scale, but we're missing out the middle parts of behavior and lifetime changes like development and aging in these individuals. So it's like, we're talking about the sandwich, but we only have the two bread slices, but there's nothing really in the middle that can make it interesting, So the open-endedness, for some reason, is not at the top or at the bottom of these time scales, but it's something that's missing in the middle. That's just something that I want to propose.
That's a very interesting quote. Takeuchi-san, do you want to say something about this?
Yeah, it's a really difficult question, but my question is, small scale and big scale is always different, and in an aging phenomena, we see the small phenomena in a cell, it happens in like a telomere shortening. But we think usually cell has a relationship, then they create community and talk to each other and then behave some kind of humanity, not humanity, it's like biology behavior. But we cannot see the relationship between smaller effect and a big like society effect. So if we find something relationship between there, like you said, like in the middle, maybe will be one of the answer for the elucidate the aging question. This is my.
That's very interesting. Yes. So we don't have aging in these systems, and the one that here showed is very nicely showed. It's static in the end, right? It's just building up these structures and either they exist or it doesn't exist, but there's no slow deterioration that you might want to call aging. So maybe aging is specific to more like multicellular communities, and in that sense, it's almost like it's like a relational property of the interactions between all of these communities. Between all these individuals, rather than a property of the individuals themselves.
Yeah, I totally agree with you. So the youngness or the aging is a property of the collective of cells, not single cell, but not the infinite amount of something. So it's the property of ensemble, which I think is a very interesting idea. And then that's exactly what the Takeuchi-san and we've also been discussing. That's the property of the collectives. So coming back to Ichihashi-san's experiment, so you said your RNA sequence is encapsulated into a droplet. That's one of the secret why they can survive, right? Comparing with Spiegelman's original experiment. So maybe this droplet can interact with each other or something, and then making like a society. Then maybe something different will happen. That's what Tom and me and also discussing. Maybe that's, and then also maybe more robust or maybe more like homeostatic. I don't know. That's
recently the droplet interact. Yes, it's interacted. Sometime I mixed all droplet and then droplet repeated the fusion and division each other. So the contents are mixed almost completely at each time. At each time means every 2 or 3 hours I mixed. So I didn't understand the point of your question. How do you plan to interact with the droplet?
Well, first of all, from my experiment with Martin Hanczyc, that we have a droplet moving around, and then we put more droplets in the system. Then there's a convection flow inside is going on, and it's randomly pointing out. But once they are interacting, this convection direction is aligned. So it's like a void model that they always convection from in this side, and then this collective motion seems to emerge in this ensemble. So maybe this collective behavior I don't know whether this this kind of order, like a big chick order parameter kind of thing, does something to do with this aging behaviors or something. But I still have some kind of hope that this collective behavior is feedback onto the micro level and then changes the property of replicators.
Yes, that's a great idea I have ever think about. Yes, if you see Martin's droplet system, droplet can move and may produce a collective behavior. It's possible. Possible.
So it's moving is very important. Like Tom said, self-moving elements is one of the key issues. I still have a hope that this collective behavior is emerging by if the elements can move around itself and then can aggregating each other and then making some.
That's true. It's interesting.
Olaf, do you have any ideas about this? Olaf?
Yeah, well, I'm really glad we're very excited all about the same thing about open-endedness, and there are many ways to create it, I guess. So I like this concept of thinking about how death evolves. So the evolution of death, not only the evolution of life. So I'm thinking about behavior of collective systems, collective animals like bees, where they have what we call a polyethism, which is old bees are going to do all the dangerous stuff,
right? And
the younger are going to be more preserved, and there is a specialization of each individual. So this interaction, not between two individuals, is what we are talking about now. The interaction between the individuals and the swarm or the collective. So this interaction between two scales, like Tom said, maybe it can be anything like time, scale, or maybe space.
Yeah, introducing spatial tempo, middle scale might be very important for understanding this at open-ended evolution.
That's right.
So here's a movie from Martin Hanczyc. He has a message on how to evolve his one of his RNA replicators. So can we see this his message? I think he's also mentioning about this the role of parasites, which is spontaneously emerging from the host sequences.
I'm thinking from, I guess, of course, from the perspective of artificial life, but my background is in genetics and evolution, and I studied quite a bit of biology, and I didn't study directly virology, but there was a point in my PhD thesis where we were evolving small RNAs in vitro in a test tube. And what we found repeatedly were these truncated, smaller versions of the RNA, which had evolved spontaneously and were acting as parasites in the system. And these were so persistent that you actually had to take steps every day in the laboratory to basically eliminate these from the system that's evolving. And because if you didn't, they would just take over. And it was interesting that when we were studying the process of evolution, which we basically wanted to show the evolution of function and show how RNAs can evolve from one state to another state and how a function evolves along with the sequence, we were, as a byproduct, finding that there are these parasites that are continuously being generated. And this seemed as a perfectly valid way for the system to evolve. Looking into this a bit further, we also can see in nature that there are certain kind of viruses that evolve truncated versions of themselves. And so they become these kinds of defective interfering particles. So basically viruses can also generate viruses themselves, which compete for resources. And it seems like this is a common element of some kinds of biological systems. And in my opinion on this is you find the evolution of these kinds of parasites, which could be viruses, when you have a system that has pressure to replicate quickly. If you have some sort of reason that the system needs to replicate quickly, The system often could find a way to form a smaller version of itself, which is not complete. So if you have a genome, you have this genome that is now shortened. And the reason that it's in the system and becoming successfully replicated is because a shorter genome replicates faster than the larger genome. It's simply a matter of this principle. So when you have a system that's under pressure to replicate fast, then it seems like these small virus type parasites tend to evolve spontaneously, whether you want them there or not. So I think there's a certain kind of system, biological system, which can produce these readily. And like I said, some of these viruses evolve naturally, their own viruses that compete for resources. So viruses are very much a natural byproduct of living systems. That's what I would call them. There's always this, fun debate that you can have with people whether viruses are alive, because then that leads to another question of what does it mean to be alive and all of these things. But I think that you don't have to restrict yourself to that kind of argument. I think you could also consider that some things are just a product or byproduct of living systems. And that's what I think viruses are. I think they're a natural byproduct of the process of life.
Okay, so I think Martin's message was very clear, and then which is also following our arguments. Maybe he's listening to what we're saying. By the way, this was recorded in August, so it's never happened. By the way, so I think Martin's idea is that host is making parasite, and parasite is making another parasite, which is virus is making another virus. This hierarchical structure of the virus is very interesting, and then one of the mechanisms that we haven't been able to find in computer simulations still, I think, right? So I think we slowly getting into the idea of a symbiotic view of life, because following our discussions and also listening to what Martin says right now, is that maybe we are not just living by ourselves, but we also unconsciously or consciously interacting with viruses, other replicators surrounding us, and maybe they are affecting us, in some non-trivial way, right? So I think it's very interesting, and then also very much adequate to this coronavirus era, right? So Takeuchi-san, do you have any? I think your presentation idea is about symbiotic view of life, especially how these microviruses can affect.
Okay. Yeah, actually, invasion or parasite is actually happen in our body, as you know. So maybe it's easy to image for the audience. So I can tell something about our body, and maybe it may be related to the evolution. So around 80% of our genome information originates from viruses infecting us and leaving their genome inside ours. And a really weird thing is that we probably wouldn't be alive without one of these ancient viruses inside us. Much of the ancient viruses in us have turned to DNA fossils. These viral fossils are buried in our DNA, transferred, but parts of these viruses are still very functional and in use. In fact, our cells have taken some of genes from the viruses and turned them into our own tools. So very interesting information. Can I show something? This is what I want to talk. Placenta. Actually, placenta is made by virus in our body. So, which means what part of us is virus. So I think I can say another way. I think virus is involved in our evolution, some kind of part of. This is a very great work written by Matsui-san in Riken. He's dermatologist, but he studied also evolution in the skin. And our ancient is evolved from fish to mammal, and our skin is also parallel to evolve. So we need to separate outside of our body from the environment, such as dryness. So we need to evolve our skin for protecting the dryness. And interestingly, very interesting thing is, as you can see here, a very important protease which can create our barrier, moisture, subspace, is also come from viruses. So if we don't have this chance, if our ancient didn't have the chance to infect this virus, we never happen. So this is really interesting. And we also live with virus, not virus, bacteria. I think maybe this is very, famous, popular information. Our body is consists of virus. Just only 10% is our gene, and then another 90% is virus. Not virus, bacteria. And interestingly, this is usually talk in the term of the gut, but our skin is also have a virus, not virus, bacteria. We found the Staphylococcus hominis, and this is exists in our skin, and this control our skin, and it change our skin to the kind of beauty like agelessness. It reduces wrinkles, skin wrinkles, and it reduces skin pores. So maybe this is kind of way of not symbiosis, but kind of symbiotic. This is actual thing. We live with another organisms, and we get some kind of gift in another living organisms. That's all.
Well, thank you so much. I think it's very interesting about still, I do remember when I was in Los Alamos in 1990 or something, I simulated evolution of host and parasite. And then I tried to find out the condition which allows the host and parasite to coexist, right? Mm-hmm. And there is only a small range of mutation rates that allows host and parasite to coexist to each other. So I think there is a very small parameter range that enables system to coexist, right? So I think this is one of the reasons why it's so difficult to have a homeostatic replicating systems. Mm-hmm. And it just somehow is succeeded in making such a homeostatic replicating and evolving system. But still, I think it's probably we have theoretically also that we have to find out what's the most important part of it. And like Takeuchi-san just showed us that we are just, assembly of a bunch of different types of species. And I do remember at that time when I was in Los Alamos, there was the famous book came out from Lynn Margulis. And mm-hmm. Lynn Margulis says that the symbiotic evolution is a critical thing, right? Before Margulis, that people think that this each species or each sequence has a mutation, and they change it into other sequences, and then there's a selection on top of it. Then that's what different speciation, different species came out, different properties came out. However, the Lin says, one species is fusing with each other and then becoming a new species, right? Like a Spirulina is prokaryotic cell is fusing together to eukaryotic cell. And then I was, I thought it was a crazy idea, right? Because there's an immune systems and they don't like to be fused from each other, right? And then from a very much, layman's idea, like why not, fusing each other, right? It's much easier. Why we have to fight against other parasites to coming in, invading us, right? We can just easy to combine everything together, right? And then we can make a big species, which is much easier. And I wouldn't. Why we have an immune system? Why we have a self-identity in mental and also physically and mentally, right? And that's a huge problem. I mean, to me, it was a big problem. So I think somehow the evolutionary system is go beyond this immune system's barrier and then fusing each other to make a new step, right? Goes to the new step, another step, and that's one of the evolutionary engine that natural system has invented, I think. Although it's difficult to make such systems in computer simulations, but that's Yes,
one thing I learned from his presentation is also that open-endedness in evolution appears when the environment is open-ended, right? So we saw very nicely the transition from an aquatic environment to the coast to the dry land and how the skin has to adapt to these different changes. Whereas all the other systems we've considered so far are closed systems in that respect. The environment background is actually stable in all cases. Whereas in the natural environment, as we just saw, evolution occurs with a very unstable background condition. And so that puts pressures that we seem not to be able to replicate in our artificial systems.
That's a very interesting point. Like Artificial Life Conference in Montreal, one of the keynote speaker is from MIT, and then she was talking about we have to think about the new materials for the robot, right? And usually the robot has one kind of a hard shell, which is we can't expect anything like what just Takeuchi-san explained. Those materials never evolving, right? The robot should survive in the same kind of hard shell, and the neural network is only they can change as time goes by, right? So I think that's maybe we should think about a new kind of materials when we make a artificial robot or artificial replicators, and how they can interact with the open environment, like Tom said, right? The environment itself must be diverse enough to let replicators to go into this open-ended evolution regime, I think. So we almost come close to 1 hour. So Hiro, what do you think about this homeostatic? Because as you know, everything is about homeostatic, right? The system is evolving, but at the same time, we have to be homeostatic, right? Otherwise, you can make something very interesting property, but it's vanishing at the same time, right? So how to get the homeostatic stable but unstable at the same time, like a chaotic system? We have to have unstable directions, but at the same time, we have to have stable directions. That's why the chaotic system, as a metaphor, it's very important to understand evolutionary systems, right? So I just want to know your opinion about this.
Yeah, if I know the solution, then I will implement that into my computer simulation and finish. But I just want to explain. Homeostasis is the ability to dynamically maintain the states at a certain level, or an ability to maintain the self. And then there is a similar concept called attractors in dynamical systems. But that attractor is a stable state, and then that state can go back to their original state if there is a perturbation from outside. Always it's stable, so it's guaranteed to coming back to the original state in the attractor sense. So it's all stable in all directions. But the homeostasis is not stable, and the states are maintained actively or in an aggressive way. So it can be stable in some direction, but it can be unstable in a different dimensions, I think. And so...
Okay, so we have to get rid of this idea of dynamical systems. So we have been, so much, affected by the ideas of attractors. The systems obeying the dynamical systems go to their attracting state. However, what you said right now is, okay, attractor normal, explains how this evolution system works. We really have to get another concept, another ideas. Don't use dynamical systems. Maybe we need some other systems like we have to think about open environment, how the system can interacting nicely with open environment, and dynamical system itself must be evolving. So maybe we have to say like a meta dynamical systems. The dynamical system is generated from these meta dynamical systems. So what we have to think about is not the dynamical system, but to think about the meta dynamical systems, right? Maybe that might be explain what you just like to say, right?
Well, yeah, it's close, but I cannot agree that we need meta dynamical systems. But still, I want to stick on the dynamical system, but I want to make more the concept of the homeostasis, and then that's very important. So the evolution is not optimization, not like attractor dynamics.
Yeah,
always need unstable dimensions, and then try to find a way to sustain self. That's evolution. That's my comment.
Maybe Olaf want to say something about this, right? Because it's very much related to what you have been doing, I think.
I guess the work on the evolution of cooperation, that was the core of my thesis. So I guess it's difficult to identify in real life, not in the model, where is the unit of replication or what is the unit of selection. And Richard Dawkins used to think that there was this selfish gene, and this was a huge idea that maybe slowed us down a bit for understanding really what is life. And instead now we know that everything is a unit of selection. Everything can be considered as this important unit that is being replicated or not, or cooperating or not. So I would consider maybe, what do you think? Maybe everything is a parasite. And the first replicator is a parasite on the environment, like Tom said, the environment of physics, just the laws of physics. And then other parasites are parasites on top of those parasites. So the environment moves one rank up somehow. So when you look for the unit of selection and the environments, actually they are everywhere. They're just at two ends of each interaction. I found this like a funny thought.
Olaf, one thing that I think we should differentiate here is maybe so you talk about cooperation and we heard about symbiosis and the parasites. So what's the difference between two species interacting a parasitic way or symbiotic way? So it seems like the symbiotic way is one where there's a mutual benefit or a gain of function that benefits both of the species or entities that are interacting. Whereas in the parasitic case, it seems to be a more one-directional situation. I wanted to say some final words also about Martin's contribution and related to this. And that is that he talked about competition between replicators and that this adds pressure on the replicators to become better replicators. So it seems to me that this pressure to become a better replicator in the long run seems to be better realized by symbiotic pressures than by parasitic pressures. Because in the parasitic pressure, it's only a one-way benefit. So the host will always lose and therefore make it less likely for the parasite to be transmitted. But if the host actually benefits from being parasitized or invaded by this other entity, so that both benefit, then it's more likely for this coupling between the species to be transmitted down the generations. And I think this goes back to what Takashi was saying in the beginning, that maybe some of this artificial life research can be relevant for understanding the pandemic. So we're all going through a second wave of the virus now, but this virus is not as lethal as the first wave. Somehow the symptoms are less pronounced. And yet it's a better replicator because it's actually spreading faster and more people are getting infected. If the host dies or if it gets disabled too fast because you get sick and you have to stay in bed or go to the hospital, then there's no opportunity for replication of the virus. So for a virus to have no symptoms at all, well, then it has more opportunities to spread itself. So ideally, actually, we're seeing a trend towards coexistence with this virus where by trying to limit its spread, we're forcing it to become less symptomatic to some extent.
I'd add to that it's a very important concept, of course, but the fitness of an entity is actually very difficult to measure in real life. So in simulation, it's just what is going to survive. You can just look at n steps, a certain time in the future, and that's how you can kind of see a posteriori how something has been parasitic or has been cooperative towards the organism, yourself, or this entity you're looking at. But actually, it's not so clear. If you look further away, maybe what the Takeuchi-san mentioned about the placenta, or something that looks like a parasite, but actually on the longer term is good for you, is very difficult to identify. So I just want to bring that, like when we look at things like the pandemic, looking at further effects like on the economy, on jobs, etc., is completely different than the short time scale. It's very tricky.
So we'll have some questions. One is whether we can symbiotic coexist with the coronavirus. And listening to the discussion that you guys saying that it's possible. And then what's the difference between symbiotic and parasitic? When you say parasitic, does it also mean symbiotic? So Tom Ichihashi-sen.
So it depends on the time scales, right? So like Olaf said, we can imagine that because of the effects of the virus right now, maybe there will be changes in our economy, and some of the effects on global warming will be reduced because there's less traveling, there's less industrial production, things like that. So maybe that can have some long-term benefits. But generally, the difference between a parasite and a symbiotic relationship is one of benefit. And whether the coronavirus can eventually actually give us benefits more in the immediate time scale, I'm not sure, but at least it would become more neutral in some respect. Maybe there is something in between being a parasite and being a symbiont where it just makes no difference to either, so it's just coexistence, but it doesn't negatively impact you, but it also doesn't positively impact you. I think in the short term, that would be the ideal scenario. And the way to get there is still actually the same as we're doing now, using masks, keeping social distance, trying to isolate, because as we saw, we need pressures in order for the system to evolve. If we don't do anything, there will be no evolution. But by manipulating the means of transmission, we're actually adding pressure on the evolution of this virus, and thereby forcing it to become less problematic for us.
Ichie-sensei, if you have any comments on this.
Yes, I totally agree with Tom's idea. And to add, I think that symbiosis, coexistence with coronavirus is inevitable, I think. It's, as Martin said, parasitic entities always appear. And now human beings prevailed entire our Earth. So we live very closely and get together. So it's an ideal host for viruses. So I think that this coronavirus is not the end. New viruses would appear repeatedly. So I think that we have to change. In other words, we have to co-evolve with virus things to live longer.
So Takeuchi-san, you have any comments on this?
This is a really difficult question, but I think we need violence actually for evolving in the future. In terms of organization in the human society, but we need to avoid death from virus caused by virus. So, which means one part we need to avoid, like over information, something, but we don't need to avoid infection. I think this is my idea.
So, Hiro, of course, you have comments on this.
Yeah, I agree with the Takeuchi-san comment. And when I'm doing the computer simulation, I can have a God's point of view. So I can observe the behaviors in short time scale and also long time scale. And I can say this is symbiotic or cooperative interaction, and this is competitive. But the coronavirus, that's our problem. So we exist in this world. So to protect myself, I just wear a mask or something. So from the God's point of view, of course, it can be a cooperative or competitive. But to protect my life or the friend's life, so we have to protect. So we need to have an individual view and sometimes have a God's view for us as a scientist.
But the nice thing about what you say here is that even though your decisions are local, of, for example, limiting social contact and transmission, the effects can be global. You're actually influencing the way in which the whole system is evolving because we're all doing similar kinds of local behaviors. And that's something that we can nicely study with the artificial life models.
Yes, exactly. Yes.
Well, actually, this is a wrapping up session, right? We have 4 different sessions from the morning. We need something more, conclusive. It doesn't have to be. We do have a conclusive messages, but this is one of the opportunities that we can sell the ideas of artificial life to people, right? In Japan, every morning, right, the people are talking about, using a SIR model, that epidemic is growing like this way, and they don't talk about any agent models, or they don't talk about any, ideas about evolution from artificial life. And then, especially, I'm very interested in Tom been doing life-mind continuity conference or meetings, right? So this life-mind continuity is a very, very important. I think it's in the spirit of artificial life. So virus and life replicators, it's not just only about this cell, but we do have to think about life itself, right? And then also the mind, right? Whether the question is, of course, we don't know whether the virus has a mind or not. But so what's the mind? Usually the people studying brain and mind is this region, and then people studying viruses and cells is here, right? But actually, what we have to do is linking from here to here, and that's what Francisco Varela, he's the philosopher behind artificial life, and life and mind should be discussed at the same time. And when I asked Francisco why you study cell, and then he said he wants to understand mind. And when he was giving a seminar on consciousness, and then he said, but I really want to understand the single cell behavior. So that's his answer, right? So what's the connections? What's the linkage between life and mind is something that we really have to understand, right? And the first, the session was about AI and AI from artificial life. The huge difference is what? Of course, we're not just talking about mind itself or life itself, but we try to link between life and mind, okay? And the second session was about Earth from artificial life. And why we have to discuss Earth? Because Earth is all about, mind and life, right? So Francisco is also interested in Earth. And then there are a bunch of, it's that the population of interested in art is growing in our artificial life community. It's because, we're not just interested in just complex chemical something, right? We're interested in what is life and then how it's related to mind that we have, right? And then third session is about, this is VR, right? VR and AR, or we call it an XR, is we are extending a reality. The sense of reality is now hugely expanding, right? It never happened in last decade. This, the changing the reality will change our way of understanding what is evolution and the way that we do computer simulation, right? So this final session, why I named it, a symbiotic view of life, is it's all about life and mind. So I really want to, from Tom, because this one is also one of the promising branch that we have to follow from the artificial community. Why this is important? Why you have to, these are not separated independent areas, but we have to integrate them. That's the final, the goal or the purpose of why we do artificial life, and then why this community is still going on now.
Yes, thank you. Well, that's quite a turnaround towards the end of our session. Looks like you had a very fun day, and
oh yeah,
the way I would.
Totally.
That's the way ALife should be done, like lots of crazy ideas, put them together and generate some new insights. So the life-mind continuity, you said technology was discussed and how VR is changing. So of course the pandemic is also making that much more acceptable and pushing us to explore these new technologies in ways that we didn't think was as possible. So maybe that's also a kind of symbiosis, right? So our coexistence with our environment and the things that we're creating, and also our technologies and social structures have homeostatic properties. They can evolve, they can adapt to our pressures and so on.
Mm-hmm.
So we also need to understand better how to coexist with our environment, such that sometimes the friction that we experience by living in this kind of environment we're creating can be reduced. But just kind of like zooming in the scales a little bit to a single organism, as you said, maybe going all the way to a cell. And I would agree that maybe we should draw the boundary between the cell and the virus. The virus is missing some important properties, such that it doesn't have its own metabolism. It doesn't have any adaptive behavior in its environment. So it doesn't seem like mind can be attributed to a virus. I would argue we can debate it. But once we are at the level of an organism, I think then the concept of mind also becomes applicable. And what's really fascinating for me is that we can actually bring bacteria and viruses back into the story. So we heard from Hirotaka-san about the fact that in our body we have all kinds of bacteria and viruses coexisting.
Yeah.
And it's just very recently that we're now understanding that a lot of those bacteria and viruses can affect our mind. So, the bacteria that live in our gut can actually produce neurotransmitters that cross the blood-brain barrier and, affect our social responses, or, make us crave certain kinds of food more because, the bacteria love that kind of sugar, something like that. So actually some of the experiences of being a person and saying, hey, I really want to eat that piece of bread over there. That's not us. That's the bacteria talking through us, right? So I really think that in the future, we're going to see a lot more of that, where we start to realize that the concept of mind needs to be rooted in life all the way down to the microbes living in our body.
Mm-hmm. Mm-hmm. Well, that's a very interesting point. And actually, in the first session was about robotics and then deep learning. And then Kuniyoshi, one of my colleague in the university, that he's making a getting up robot, right? Waking up robot. And then his remark was, we really have to, put the gut feeling into the robot, right? And then the gut feeling determines how to behave. And then I really, I totally agree with him. So I'm discussing with people in the UK that they are good at, making a robot, which is, eating snails and get energy from it, right? So why not, putting my Android, the guts into my Android, and then once he gets, starved, and then he does something else, right? So the free will is not from our brain, but maybe it's from gut, like Tom said. And I really like this kind of idea. And then we do have to change whether this neural network is all about our mind. Maybe we have to do some other kind of computational modeling to understand how the mind works. So maybe the one with the gut is one of the good idea that we really have to bring it to a computation model. Maybe the hero is laughing, so he's not agreeing with me. But what do you think?
Well, actually, I like the idea, God's idea, but at the same time, I'm giving up this kind of discussion at the same time.
What?
No, because I'm always creating the non-life things, and then try to understand as an outside of what I created. So what I can create is not life, but I have to keep creating the life. Try to create the life, but it's a non-life. Then the actual life is outside of what I make. I think that's my stance.
That's your obsession, I must say, right?
Can I say something, Hiro? I think even though you think that you're failing, I think your attempts are really valuable, because really by trying to create life and failing all the time, or in general, what Olaf and Norikazu-san said about trying to create open-endedness but failing, that's how we learn more about, how is it really happening in the natural world. And by never running into these walls, we would never realize that there's still something that we don't fully understand. So I think all these failures are actually successes in us realizing that there's still much more to learn.
Yeah, exactly. In that sense, the gut approach can be a failure. And go to the
what? Yeah, but as Tom said, it's a creative error, right? Once you don't make an error, then you are not taking risk. And we really have to take a risk to make an error. And that, makes new kind of products in the future, right? I think but that's all about, our life and research. So it's almost 5 minutes, then we have to wrapping up this session. I don't know what kind of message that we have to bring up from our session, but this is kind of mainstream of artificial life, right? We, back in 1987, that we started to discuss what is replicators, and then what we can, computationally enable to have replicators in a computer, and of course it's possible, right? And I remember in 1989, when I was in Los Alamos, the mainframe was stopped because there's a bunch of viruses, computer viruses is generated, right? I was the first time that the computer stops. And everybody was there, and then people talking, hey, this computer doesn't work because there's viruses in there. And I was surprised. What are you talking about? And there's a bunch of viruses from somewhere in Maros, and then it's sucked over there, right? So artificial life was already there before the artificial life started, So that this computational viruses is so important to understand. And then after that, like Hiro explained, Chris Lampton's loops came out, and Evoloop came out, and then there's a bunch of different types of replicators it's invented, but still we don't understand what's this open-ended evolution can take place, right? And then this challenge is still going on. I don't know whether still we can get $1 million if we can successfully invent open-ended evolution in computer or whatever, right? But that's what that's our that's community's obsession, that to have an open-ended evolution in either computer or in real life. And then from this session we're finally discussing this life-mind continuity and symbiosis. So symbiosis, I think, is one of the way to me, it's the link between life and mind. So when our body is linked with viruses or the replicators, it's not just a physical entity, but maybe like mind is also, contagious from outside. That's kind of a recent idea that I have, right? When I make an Android, of course, doesn't have a mind. But once the Android begin to interact with humankind, right? Then mind is slowly copying from human to Android. So maybe the consciousness is also contagious. That's also I'm discussing with Martin, that consciousness can be contagious, right? So if you are living by yourself without interacting with each other, right? Nobody's there. There's no mind. That's kind of my hypothesis. So parasite is not virus evolution. It's not just a metaphor, but I think that's also happening, and then that's what we have to understand to further going on to follow this idea of life-mind continuity. That's kind of my concluding message. So thank you so much for joining this session, and let's keep in touch and discuss further. Thanks so much.
Thanks.
Thank you.
Okay.